Hello everyone, my name is Cedric and I'll be your host for today. Welcome and thank you for taking part in MDPI's webinar entitled Air Pollution and Biomarkers. This is the fifth webinar in the toxic series, especially our journal of the same name. If you are interested in contributing to this topic, don't hesitate to submit your research. Simply check the webinar's webpage for more information. I would like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the webinar's webpage. Please note that you will have to be logged in to SciForum in order to view the recording. Certificates of attendance are available to the webinar attendees on their SciForum profile. To access yours, visit sciforum.net, create an account if this isn't yet done, then simply log into your account and go to My Certificates. There will be two presentations today of 30 minutes each. The presentations are followed by a question time moderated by the chair. If you have any questions for our two speakers, please post them in the Q&A box at any time during the webinar. You can find the Q&A by clicking on the Q&A option in the Zoom menu bar at the bottom of your screen. Please use the Q&A box and not the chat panel, and please remember to add the name of the person to whom you're addressing your question. Today's webinar is chaired by Professor Doug Brugge from the Department of Public Health Sciences, School of Medicine at the University of Connecticut in the United States. Doug Brugge is a professor and chair of his department, and he has been principal investigator on a series of NIH-funded research studies of ultrafine particles from traffic and their association with cardiovascular biomarkers. Thank you so much, Doug, for chairing today's webinar. And that's it for me. I now hand over the microphone to our chair, and I hope you all enjoy. The floor is yours.
Okay. Thank you, Cedric. A good way to get us started. Can everyone see the screen that I'm sharing? Cedric, maybe you could confirm.
Yeah, we are on full screen. Yeah.
Great. So it's a pleasure to be here this morning, and it looks like there are attendees from many places around the world, which is quite nice. I present a lot in the United States and sometimes at international conferences, but this is the first webinar that's truly international that I've been doing. The session today is Air Pollution and Biomarkers. It arose out of a special issue that I am co-editing with Dr. Mei Chung from Tufts University, also in the US, on the influence of airborne particulate matter on biomarkers and biological pathways. And if you have research in that area and you are getting close to being ready to publish, we're extending the deadline to the end of April, and you could consider submitting to this issue. And I would encourage you to do that. I'm going to talk about the core area of work that I've been doing for the last 15 or so years, traffic-related ultrafine particles, cardiovascular biomarkers, and protective effects of air purifiers, which is our latest line of research. So I think many of you probably know, but I actually don't know much about the audience, so I'll just explain everything. Particulate matter in the air is classified by size. It could also be classified by composition, but it's much harder to do, so we usually stick with size classifications. This is a graph that we've used a lot from the US EPA. This is representative of a human hair, 50 to 70 microns in diameter. And then these blue circles are PM10, the largest of the regulated particulate matter, which is mostly dust, pollen, mold, etc. And then these small red circles are PM2.5, which is the most studied ambient particulate matter, and is usually from combustion or secondary formation secondary to combustion products or air pollution. And that is a very well studied, there's still a lot of ongoing research on it, but there's a lot already known. If you take one of those PM2.5 circles and you enlarge it, then these little black dots would be the ultrafine, the largest of the ultrafine particles, which are 100 nanometers and smaller. And there's much less research about ultrafine particles. And that's why we got into it and started doing research in this area, which we've been doing for some time now. Ultrafine particles, unlike PM2.5, vary much more dramatically in space and time. PM2.5, being largely a secondary pollutant, tends to be sort of spread broadly over large geographic areas, hundreds of kilometers, whereas PM2.5, and if you look at the red and the green dotted lines here, and you look at, sorry, I have to move my the panel with my video in it. If you look at the red and green lines here, and if 0 is the edge of a major roadway, the ultrafine particles drop off rapidly. And we've looked at this. This is an old graph. It really should be updated. There's been a lot of research since then. But generally, it's accurate that out to 200 or under special conditions out further, you'll see this gradient of ultrafine particles on the downwind side of major roadways. So that means that people living near major roadways, people driving on the roadways, people spending time there get a high exposure, and people further away get a low exposure. So there's a very fine-grained spatio-temporal pattern to the exposure to ultrafine particles. And I don't know for China, Dr. Zhu, and maybe you could say something later, but in the European Union and in the United States, road transportation is one of the top 2 sources of ultrafine particles. Our interest in terms of biological pathways is illustrated very well here. This is from the US EPA Integrated Science Assessment for Particulate Matter that was published recently. Here's the ultrafine particle exposure. And one of the pathways we're interested in is the activation of the autonomic nervous system, which affects blood pressure, and blood pressure is one of our primary outcomes. The other pathway that we've spent a lot of time looking at is systemic inflammation. And both of these go on to cause or contribute to serious heart outcomes, including ischemic heart disease, myocardial infarction, stroke, and so forth, cardiac arrhythmias. But we're more at the biomarker stage right now. We're trying to get into this area more, but up till today, and what I will present today is about these intermediate biomarkers on the pathway toward overt disease. One of the other things that is concerning about ultrafine particles is that they cross biological barriers. And this is work by Lilian Calderon-Garcia-Duenas, who's in University of Montana. She contributed this photomicrograph to a book that I co-edited with my former student, Christina Fuller, now at the University of Georgia. And this is a photomicrograph, an electron micrograph, transmission electron micrograph of brain tissue at autopsy. And you can see these ultrafine particles embedded in the brain tissue, in this case in a mitochondrion cellular level. And these particles, I feel like she proved 2016, 2017 proceedings of the National Academy of Sciences have a morphology that shows that they were formed at very high temperature. So they're not something that formed inside the brain. You don't have 400-degree temperatures inside our brains. So they formed in combustion outside of the body, and then they got into the brain, and probably by traveling directly through the back of the nose, up the olfactory nerve, and into brain tissue. The focus of our work has mostly been epidemiology rather than toxicology. And my colleague, Mei Chung, who I said was co-editing the special issue with me, did a scoping review through 2017 that was also published in our edited book about ultrafine particles and health. And this just shows the pathway of selecting and the studies to review. At that time, at the time of her review, there were 97 studies that she included in her publication, again in our book. But and there was another review around the same time. There really needs to be an additional review because since 2017 there have been a lot more publications. But this will give you an idea of what the state of the literature was at that time. And what you can see here is on the x-axis, we have year up through 2017. And then on the y-axis, we have different health outcomes. CV is cardiovascular, R is respiratory, and then other. And then green means there was no association found. Orange means there was an association that was not statistically significant. And red is a statistically significant association. And then this chart here is all short-term outcomes. That is hours or days or maybe weeks following exposure. And what you can see is there are a lot of short-term studies. There tend to be many more analyses. Each of these circles is one analysis, not a full paper. So an individual study might have multiple outcomes. And then the size of the circle is the sample size, the number of participants. So smaller circles are smaller studies, large circles are larger studies. This is a graphical approach that May pioneered and has used. And what you can see is that there's more studies of cardiovascular and then respiratory and then less other studies. Although I would guess if we extended this out, we would find a lot more studies of neurological outcomes subsequent to 2017. At that time, there also, if you look over on the right-hand side, you will see that there were very few studies of long-term outcomes, which is usually defined as 1 year of exposure. And our research is in here, published in 2016, and there have been more studies since then. So there is more, but compared to PM 2.5, compared to short-term, long-term exposure studies of ultrafine particles are rare and not very common. And that is a serious limitation to the possibility of regulating exposure to ultrafine particles because the US EPA, and I assume agencies in other countries as well, don't feel like the evidence is strong enough to warrant a separate standard for ultrafine particles. So in our research, I'm going to describe our first major study in a little bit of detail. What we did is we were in the Boston, Massachusetts area. These are two major highways here, I-93 and I-90 that go in opposite directions, north-south and east and west. And then along these highways, we chose neighborhoods that were near the highway and then further away from the highway so that we would have a population of near highway residents and a population that was what we defined as urban background. We built a mobile laboratory that measured ultrafine particles as particle number count using condensation particle counters, and then drove it around each of these neighborhoods many, many times over the course of a year, each paired neighborhood for 1 year. So it took 3 years to complete this work. And Alison Patton, who's now at the Health Effects Institute, did her PhD thesis on the air monitoring and model building. And what she did is she built models of ultrafine particle concentration, particle number concentration, technically. And you can see the combined annual average of those outcomes for the neighborhoods here, with blue being low concentrations and red being higher concentrations. And you can see near the highway, the concentrations are much higher than further from the highway. Boston, Chinatown, is right at the intersection of two major highways here. But this doesn't really tell you everything. These models were not just an annual average. They had an output for at a 20 by 20 meter resolution for every hour for an entire year. So in each of these neighborhoods, at every 20 by 20 meter grid, there is a separate value for every hour over the course of an entire year. And we did that in order to try and capture the rapid change in space and time of ultrafine particle concentrations. The other half of the project, so that was the air monitoring model building. The other half was recruiting participants. So each of these green dots is a participant that we recruited into the study. And they are located in the neighborhoods where the models were built. Early on, we realized because of the very fine-grained nature of changes in ultrafine concentration, that we had to be very precise with our assignment of geographic location of people's residences. And if you just do address mapping, GIS address mapping, you can have large errors to that. So here's an example of a residence that is 85 meters wrong from where it's supposed to be, where it actually is. Here's one that's 90, 95, especially in these larger housing complexes. There tends to be one address for the complex, and then the actual location of the apartment is somewhat different. It can be substantially different than that. So Kevin Lane, who is the other graduate student, Christina, who I mentioned earlier with the book, Allison and Kevin were the three graduate students who did their PhD theses on this. Kevin is now an assistant professor at Boston University. And Kevin manually moved every address to the exact location of the apartment in its building or on its street in order to get precise geocoding for the future analysis. We then took participants and we asked them about their time activity. And what you can see here in the top graph is people who work or attend school, mostly employed, not that many students, because we were recruiting older adults, age 40 and older. And then in the bottom graph, you see people who are retired, disabled, or unemployed. And what you can see is that the people who are employed, the blue color here is time they spend away from home at work, and red is the time they spend at work. And the retired and unemployed people spend much more time at home and are not away at work nearly as much. They also, this black line is time spent on the highway. And so you can see morning rush hour and evening rush hour. So the people who are working spend a lot more time on the highway, and the retired and disabled avoid the rush hour. And some of them go out on the highway, but it's in the middle of the day, a different time frame. And we corrected their exposures for their time activity. So for that every hour of a year, instead of just assigning the exposure to participants outside at their residence, we adjusted for how much the pollution would be reduced going inside the residence. We adjusted and gave them a different exposure assignment when they were at work, and yet another exposure assignment when they were on the highway. And this proved to be very, very important. If you look on the right-hand side, you'll see near the highway, again, red is higher exposures and blue is lower. You see in the urban background area, low exposures, and near the highway, much higher exposures. But when you adjust for their time activity patterns, the near highway exposures are reduced. And that's because many of those people are going to work, and when they're at work, they have a lower exposure because they don't work near the highway. They work in a building with good ventilation or whatever. So that adjustment is critical. There's a huge exposure error if you don't make this adjustment. And in fact, if you don't make the adjustment, you cannot see the associations we were looking for. At the top panel here, this is particle number count, and then this is just the residential annual average, the annual average outside their home for a year. And you can see there's a little bit of a trend of association with higher concentrations, with greater levels of C-reactive protein, an inflammatory marker in their blood. But when you adjust for time activity, It moves a bunch of these higher exposures into this intermediate area, and then you get a much more monotonic association that looks more realistic and suggests a much stronger association. And then the final step is you also have to adjust for confounding. And we had negative confounding. The top panel is a subset. The bottom panel is the full study population. And when you adjust for confounding, you get again this monotonic, almost linear type of association. And the problem here is that the top panel is the city of Somerville. When you add in Boston Chinatown, which I mentioned earlier, you have largely Chinese immigrants and some Vietnamese immigrants, and their association is really different with I won't go into that today, but their association is really different. And if you don't adjust for that, you can't see this association either. So I hope okay, and then the final step, and this is just in press now, it's in the Science of the Total Environment, and Kevin Lane, my erstwhile former graduate student, now assistant professor, continued to work on this and got it published just now. It just was accepted a month ago. And what he did here was structural equation modeling, and I won't go into details here, but suffice it to say that when you put the three inflammatory markers that we measured, C-reactive protein, interleukin-6, and tumor necrosis factor alpha receptor 2 into a single construct, and then you also add distance to highway to take into account other near highway air pollutants, you get much stronger associations even than those that I showed you before. So I hope this convinced you that taking an approach of highly detailed, exacting care with exposure assessment is necessary, at least in a small population, to see the effects of ultrafine particles on cardiovascular biomarkers. So that's 10 years of work. I'll just mention very briefly another graduate student of ours, Matt Simon. I'm continuing to work with him now at the Volpe Center, Transportation Center. Used his PhD thesis to develop a larger and more detailed model of ultrafine particles. This is for the city of Boston and the city of Chelsea. And his model is an improvement. Allison's model was unable to capture the nighttime dips in ultrafine exposure, and Matt's model was able to do that. He did that by adding a central site monitor in the middle of the study area, so there was a continuous temporal record of ultrafine particles, and then was able to use that to also extrapolate backwards and forwards in time what the ultrafine particle levels are outside of the year in which they were measured. And that's all been published most recently in Environmental Science and Technology in 2020. You could look that up under Matt Simon. I want to say a little more about the biology, just because I think this is a really important future direction that we would like to go in. We haven't been able to do much on it yet, but we'd like to go there. We think that the inflammation that we're seeing, the C-reactive protein, the interleukin-6, IL-6, is being generated through the NLRP3 inflammasome. So the NLRP3 inflammasome catalyzes the conversion of IL-1 beta, pro-IL-1 beta into IL-1 beta, and pro-IL-18 into IL-18, and then IL-1 beta in turn generates IL-6, which generates CRP, which are the outcomes we looked at. We did a small sub-study trying to look at IL-1 beta, and it didn't work with our blood samples. The levels were just too low. We had a lot of non-detects. But what we'd like to do is look at the inflammasome itself, and trying to collaborate with an immunologist at the University of Connecticut, where I am, and seeking some funding from NIH to do that. But it is established that other particulate matter, not necessarily ultrafine particles from traffic combustion, but other types of particles do act through this inflammasome to generate quote unquote sterile inflammation. I want to say just a couple words before I finish about our latest direction, which is working on interventions to reduce exposure and prevent adverse health outcomes. And the primary focus that we've taken is these in-home air purifiers. They have a HEPA filter, they have a fan, they recirculate the air, and they filter out many of the particulates, or most of the particulates, and then recirculate in the room. We've done 2 pilots. This is a systematic review and meta-analysis of air filter studies to date. Not to date, up until recently, up until a couple of years ago, because they didn't capture one of ours. But 2 of ours are included in this. The combined effect, if you're used to reading forest plots, the combined effect here seems to be and the top graph, I believe, is IL-6, and then no, the top graph is CRP, C-reactive protein. The middle graph is interleukin-6, and you can see that the inflammatory markers appear to be reduced in when you combine all of the studies. Our studies did not help the analysis very much. We didn't find very large reductions in these inflammatory markers. Here for IL-6 was the one case where we found a statistically significant reduction. We learned a lot from doing these small pilot studies. These were 20, 23 people each. And what we learned, one of the things we learned was that it was very hard to control for all the factors. People were sick, they weren't in the room with the filter all the time, et cetera. They were generating indoor concentrations. So we did another study that we published in 2001, early 2001, so 2 years ago, in which and this was again in Boston, Chinatown. This is a subsidized housing development right at the intersection of the highways. It has a community room, and we put 77 people in that room for 3 two-hour sessions on consecutive Mondays. So Monday morning 1 week, 2 hours, Monday morning the next week, and then Monday morning the third week. And during those 2 hours, Nilakshi Huda, who's one of the 2 co-equal co-authors here, created conditions by opening or closing windows and using these air purifiers for low, medium, and high exposures to the traffic pollution. This is, and we could see different low, medium, and high ultrafines, but also black carbon concentrations. And the really dramatic finding here, which was analyzed by Misha Ilyashu, who's our biostatistician, was that blood pressure was lower in the low concentration, medium in the high concentration, and then much higher in the highest concentration. And so this convinced us that in a highly controlled setting, we could see that these air purifiers reducing the particle concentration from the highways could influence blood pressure in a positive direction. But it was a highly controlled setting. They're wearing hearing protection to reduce sound, to cancel out sound. They're sitting still, and it's very, very structured. So the question is for us then is whether that can be translated into the real world environment. And we are in the middle of a randomized crossover trial of air purifiers near the highway, not in Chinatown this time, but in Somerville, the other city we worked in a lot. We got off to a slow start. COVID interfered the first year, but we now have over 90 participants have been enrolled, and 75 of them have completed the study. And the study is 1 month of either real or sham filtration, 1 month washout period, and then 1 month of the other configuration. And we collect blood pressure, including central blood pressure, and blood samples for inflammatory markers at the start, and then the end of the first month period, then the start and the end of the third month period. And we haven't analyzed this for the outcomes yet. We have to keep it blinded until we're ready to do that. We've also worked on with communities on landscaping and urban planning and building design, including ventilation systems in buildings, and a lot of focus on noise barriers near the highway. They reduce not only noise, but they reduce the air pollution that gets from the highway to the participant homes. And I'll just show you this to conclude. This is one of the neighborhoods in the city of Somerville. You can see the houses right near the major highway, and then this little rickety chain link fence is the only thing between the housing and the highway. And our dream, it has not happened yet, but we've been working with the community. Our dream is to change that into something more like this so that they're protected. The community would like greenery, they'd like a clear barrier rather than a concrete wall, and maybe that could transform a neighborhood like this. So this research is all on our website. There's a ton of stuff there. If you put CAFEH, which stands for Community Assessment of Freeway Exposure and Health, into Google, the website comes up first, and you can explore all our publications are linked there, our policy documents, many, many things that that might be of interest to you. So with that, I think I'll, whoops, I think I'll conclude. I see there are a bunch of chats. Are there any particular questions from my talk? Is there a set range to consider long-term exposure to PM2.5 for the onset of any health symptoms? For all of these air pollution studies, the long-term exposures most commonly are 1 year, but the best studies, I think the best studies look out over multiple years. I don't know what the longest is, but there are studies that go back over 10 or 12 or maybe 15 years, maybe more, I'm not sure. But that's a really good question, because the exposure someone received in the last year may not be representative of their lifetime exposure. And we might reasonably assume that lifetime exposure is a more important indicator of their health risk. Could we replicate your research in Nigeria? Wow, I'd love to do that. I haven't done much international work yet. I have some nascent collaborations in Sri Lanka, India, and Vietnam. I don't know why it's all in Southeast and South Asia, but it is. We haven't had any funding for any of that research yet, but I'm interested in international collaborations. It's new for me. I'm still learning how to do it, how to get funding. Like I said, we weren't very successful so far, but I'm always happy to talk. And I will say one other thing, Dr. Abdulud, I'm not sure I'm pronouncing that correctly, but your question is really on target. There was just a publication of air pollution in Africa, looking at and the impact is huge. It's not surprising. We knew there was a huge air pollution impact in East Asia, in South Asia, and now finally the focus has turned to Africa. And of course, there's a big, I've forgotten the numbers, but I think about 1 million deaths a year in Africa, similar to India and China. So it's a very important question, and I'd be happy to talk to you offline if you email me. Okay. Dr. Zhu, I think if you could put up your slides.
Okay, sure.
And I am so sorry. I know you sent me a short bio, but I forgot to get it out and put it up in order to have it ready to introduce you. And I was thinking about my own talk, and so I'm not ready to do that. But I will just say, I'll let you introduce yourself, but I will say the preamble to your own introduction of yourself, that we've never met, we haven't worked together. It's a totally new association for us to do this. And I love to make new acquaintances, to have new colleagues, and I deeply appreciate your willingness to participate in this. And I look forward to hearing your presentation. Please take the floor.
Okay, thank you. I would start by thanking for your invitation to this webinar. So I would have a chance to present some works from my group. So basically my work has been focused on the toxicology of air pollution using in vitro and in vivo models to understand the biological mechanisms about the adverse health effects of air pollutants. My focus specifically on PM 2.5 and ultrafine particles. And today I will be more talk about the cross-generational effect of PM 2.5. So this will be my today's topic. And if you guys are interested in other potential mechanisms or other action modes of PM 2.5 or ultrafine particles, we can have more communication later, maybe. Okay, so we all know much about air pollution already, quite familiar with air pollution. Air pollution was the fourth leading risk factor for early deaths worldwide in 2019, passed only by high blood pressure, tobacco use, and poor diet. And WHO already estimated that more than 90% of the current world population still live in places exceeding the WHO air quality guideline, among which, as you can know easily, China is one of the most polluted countries, with more than 99% of the population experience annual average PM 2.5 concentration over or exceed the guideline. And according to the new WHO air pollution guideline, which is 5 microgram per cubic meter. So we have already seen a big number of studies demonstrating that exposure to PM 2.5 correlates with increased premature mortality and a lot of diseases such as cardiovascular diseases, pulmonary diseases, and so forth. So except for the adverse health effects caused in the direct exposure population, for example, the adult population that is directly exposed to PM 2.5. Actually, more and more epidemiological and toxicological studies have shown that PM 2.5 may also have cross-generational effects, which means the parental exposure to PM 2.5, which could either from the maternal line, or I would say maternal exposure or paternal exposure, or both, they are associated with interrupted development of human offsprings, and this could be a risk for developmental programming of offspring. For example, this could lead to low birth weight, intra-uterine growth retardation, and preterm births in neonatals. And it's not just about the adverse birth outcomes. A focused scientific effort over the last decade already demonstrated that, the parental exposure to environmental stressors, for example, PM 2.5, they may persistently impact offspring's susceptibility to a variety of non-communicable diseases, for example, obesity, which could refer to as the developmental origin or developmental programming of health and diseases. And accumulating evidence suggests that results such as environmental pollution like PM 2.5 during the perinatal period, they could alter the developmental trajectory of our fetus or the offspring, and finally leading to long-term detrimental outcomes, which could often culminate in adult pathology. So this could be a very important issue for us. And actually, this developmental programming of health and diseases may happen via a lot of mechanisms. For example, the change in gene expression, protein expression, and one of the big mechanisms could be through the change in epigenetics, which involves genetic control by factors other than an individual's DNA sequence. Epigenetic change can switch genes on or off without changing the DNA sequence and determine which proteins are transcribed. So studies have already shown that environmental exposure to PM2.5, for example, they may harm the fetus by impairing the epigenome via, for example, DNA methylation, histone modification, non-coding RNAs, and so forth, of the developing organisms to modify the disease risk later in life. And they might also change the epigenome of the germ cells. And this change in the germ cells might pass down through generations to generation. So when we think about the cross-generation effect of the PM2.5, we will think about two sides. One is from the maternal exposure, and the other is from the paternal exposure. So if we check most of the literatures, we will find that there are much more studies starting the maternal exposure to PM2.5 compared to paternal exposure. So if we check that, we'll find, yes, maternal exposure to PM2.5, they might cause developmental programming of the obesity, for example, in offspring. But a interesting question is for us is, there are other questions, for example, what is the vulnerable windows for this adverse programming? Is this developmental programming by maternal exposure multiple generational transmissible or not? And we also want to understand what is the potential mechanisms underneath. For example, about leptin, which is a well-known hormone predominantly produced by the adipocytes that could help regulate long-term energy balance. So we want to check if this leptin signaling pathway is playing a very important role in this developmental programming or not. So in our study, when we start with the maternal exposure, we set a model using a mice model, which is a wild-type female C57 mice model. It is a very widely used model for toxicology. And we expose these female mice using a versatile aerosol concentration enrichment system. As you can see, the big version of the system you can see in the images. So this system allows for long-term whole body exposure of the PM2.5, and we just get the PM2.5 directly from the ambient. So what we can tell from this experimental scheme is that for the F0, which means the exposed generation. So we expose these female mice with FA, which is filtered air, the control group, or the PM2.5 CAP is concentrated ambient PM2.5. And after the exposure, we made them with normal, non-treated, which means non-exposed male mice, and we produce F1. So that's their F1 offspring. And then we made them with normal mice again to produce F2 and F3. So what we can tell that the level, the PM2.5 level in the CAP, which is PM2.5 group, is around 200 to 300 microgram per cubic meter. And the PM2.5 level in filtered air is around 12 to 17 microgram per cubic meter. This level actually is quite common in previous years when we were in winter in the northern part of China. So it's quite comparable to what we are exposed for population or for people. So what we did is we split that into 4 different groups: FA, CAP, of course, the control, and positive control or treated group, PM2.5 treated group. And we also have pre-conceptional and post-conceptional group. So after a 7-week exposure to FA or CAP, the mice were mated with normal, non-treated mice. And upon the presence of the sperm plug, they were neither maintained in the same exposure or switched to the other alternatively. So what we can find in the birth weight is that the exposure, maternal exposure to PM2.5 within the pre-conceptional period and the gestational period, which is the black one, CAP group significantly decreased the birth weight of the offspring. And very interestingly, we found that the preconceptional group, which means exposure throughout the 7-week preconceptional period alone only, they also significantly decreased the birth weight of the offspring, but the exposure throughout the gestational period, which is the post-conceptional group, the purple one, did not significantly affect the offspring's birth weight. And also the leader 1 and the leader 2 have very similar trends. And we found that in leader 3, the offspring from the preconceptional dams have comparable birth weight with the control group. So which means maternal preconception exposure to PM2.5 is sufficient to decrease the offspring's birth weight. And this programming capacity of PM2.5 is reversible. And when we check the growth trajectory curve and histological analysis of the adipocyte tissues, and we also find this maternal preconceptional exposure also is sufficient to program the male offspring's development and adulthood adiposity. In order to check if this is multiple generational transmissible, we check the F2. You can tell easily that one side is from the maternal line, F2, and the other one is from the paternal line. And you can tell easily that the male offspring in F2 from maternal line, but not from the paternal line, had significant lower birth weight, increased postnatal weight gain, and adulthood adiposity. Compared with the paternal line, we did not see any of this phenomena or phenotype. And we also checked F3, and we did not find any of this phenotype happened in F3 offspring. So this developmental programming by the maternal exposure to PM2.5 disappeared in F3. So in order to understand what is the potential mechanisms, we detailed a further analysis, the F1 offsprings, and we can check that. First, the PCR results reveal that the maternal exposure to CAP did not significantly change the expression of adipocyte differentiation markers. And since we know that adipocyte inflammation is very important and very strongly correlated with increased adiposity, and the inflammation in adipocyte is very important and could play a critical role in obesity-related pathophysiology. So we check the inflammation levels in adipocytes, and we find, yes, significant increase of inflammation levels in adipocyte. And we know that obesity is very common risk factor for abnormal glucose homeostasis. So we check the homeostasis-related parameters, and we found increased fasting insulin level and increased plasma-free fatty acid levels in the male offspring as expected. So next is we want to check what happened, what is the mechanisms. So consistent with the body weight, we find that maternal exposure to CAP significantly increased the food weight. And we found the intraperitoneal injection of leptin resulted in a greater decrease in food take, which means a more sensitivity to the leptin. So leptin, we know, is well known as a very important hormone in homeostatic control of food intake, and central leptin resistance is believed to be one major component of the pathogenesis of human obesity. So it's very important here to check the role of leptin in this developmental programming of obesity in offspring. So what we find is seems that they are more sensitive to the exogenous leptins, so which means leptin is still working. So then we check the expressions of leptin target genes. We find no change in all of these genes. How about the leptin levels? So what we did is we also checked leptin levels in the plasma, which is circulating leptin level, and in the adipose tissues, which is adipose leptin level. And we found that exposure, maternal exposure to PM 2.5 significantly decreased the plasma and adipose leptin levels. And another important one is, since we know that the promoter region of the leptin gene is subjected to dynamic methylation, so we then isolated the adipocytes and then assessed the methylation levels of the CpGs within the promoter region of the leptin gene, and we found significant increased average methylation level of the leptin promoter, which is specifically at site 1 and at site 10. So, which means the methylation of leptin gene could be a very important potential mechanisms underneath this developmental programming of obesity happened in male offspring, which is induced by maternal exposure to PM 2.5. So now we know about what happened in maternal exposure. We also want to check what happened. How about paternal exposure? So similarly, we do a very similar design, experimental design as here showed in the same system, but we use the male mice for exposure. So what we do is, again, we just expose the male mice under filtered air or PM 2.5, and then they were mated with normal males, females, to produce the offsprings from F1 to F4. So what we found actually is, first, we did not find any change of the duration of the sears to impregnant dams, or the duration of pregnancy, or the litter size, or the sexual ratio of the offsprings. So these parameters are not changed by paternal exposure to PM 2.5. And how about the birth weight and the growth of these offspring? So we are very interested. So we check the birth weight and the growth rate of these offsprings, and we find that paternal exposure to PM 2.5, they induced a significantly low birth weight, very similar to what we see in the maternal exposure, but they retarded the weight gain of the male offspring. So if you still remember, what happened from maternal exposure is the weight gain of the offspring of the male offspring actually are increased. So instead of increasing the weight gain, this growth of the male offsprings are retarded. And similarly, we also find the decrease in food intake of the male offspring, but surprisingly, we find an increase in adipose tissues, including epididymal, perirenal, adipose tissues, and this is very interesting. So we have decreased birth weight, decreased weight growth, but increased adipose tissues in these offsprings. So again, we also want to check if this programming of offspring is cross-generational transmitted, can be cross-generational transmitted or not. So then we just use this F1 and mated them with non-treated mice and just produce the F2 to F4 offsprings, and we can tell easily from this data. I'm not showing you the maternal data, but we did that. So what we can tell from these data is that the paternal grandfather exposure to CAP can result in significant decreases in the birth weight, postnatal weight gain of the grandson mice, and all this, we can observe this retardation of this postnatal weight gain also in the offspring from a paternal line in F3, but not in F4. This programming disappeared in F4. So this is quite different. We find that for maternal exposure to PM 2.5, this programming of offspring happened until from F0 to F2. And actually for paternal exposure to PM2.5, we found this phenomenon or found this effect happened from F0 to F3. So lasted more than the paternal or maternal exposure. And again, we also checked the adipose adulthood adiposity. And this also very similar to what we see for the gross weight gain, birth weight and the gross weight gain. So yes, we also found adulthood adiposity in F2 and F3 from paternal line, but disappeared in F4. All the offspring in F4, we did not see any of this effect. So actually, we also checked the weight, the weight of the main organs and tissues of all the offsprings, and we find the exposure, paternal exposure to PM2.5 significantly decreased the weight of the liver and kidney, but not any other tissues such as lung, heart, pancreas, and so forth. We also did histological analysis on all of these tissues, but we did not find marked morphological abnormalities caused by this PM2.5 exposure in the liver or kidney. So we might to dig into that, like what is happening in liver and kidney, but we're still working on that. So actually, I have mentioned previously about epigenetics. I want to mention specific one about microRNA. MicroRNAs are a class of non-coding RNAs that are thought to play very important roles in regulating gene expressions. In most cases, the microRNA, they might interact with the untranslated region of the target messenger RNA to induce messenger RNA degradation and translational repression. And sometimes we'll find this interaction of microRNA with, they could interact with other regions of the messenger RNA, or they could even activate translation or regulate transcription. And actually, previous studies, a lot, not many, but several, have already reported that the paternal, the microRNAs in sperm, in paternal sperms, they could be a very putative mediator for the paternal exposure-induced programming of certain phenotype. For example, here I listed some of them, like the behavior and metabolic disorders by traumatic stress, the depression susceptibility by chronic mild stress, and HPA axis response by chronic stress. So the sperms in the paternal, the sperms could carry a lot of information for the cross-generational effect, and among them, microRNAs could be very important. So from this background, we think maybe we should check what happened about the microRNAs in the sperms exposed to PM2.5. So what we did is that basically we just collected the sperms of those PM2.5 exposed mice, male mice, and we provide the sperm microRNA expressions using microRNA chips. So what we identify actually is we in total identified 11 differential sperm microRNAs in F0, which is the PM2.5 exposed generation. And after controlling the false discovery rate using Benjamin-Holberg procedure, and we reconfirming the results using PCR, so what you can tell that we only find one differential sperm microRNA, which is MMU and microRNA 69065P. And we also check the difference of these microRNA levels in the sperms of the offsprings. So what we found that very interesting, we also found this microRNA is significantly increased in the sperms of F1 and F2 offspring, but not changed. The level is not changed in the sperms of F3 or F4 offspring. So if you still remember the cross-generational effect or the developmental programming effect caused by the paternal exposure of PM2.5 lasted the effect on the birth weight and on the weight gain actually they disappeared in they still happen in F3 and they disappeared in F4. So actually this result is very consistent with what we see in the phenotype. So if we check the literature, we can find actually this microRNA is not new. It's not new for us. Actually, there are some publications reported that this microRNA 69065P, the level of this microRNA can be changed by exposure to several environmental pollutants, such as the bisphenol A. And for the PM2.5, this is quite new, but it's not very new for the environmental pollution. But how this microRNA is working as a mediator to mediate the adverse effects is unknown, it's not well studied. So if we go to GO enrichment analysis, we can find that these potential genes are mostly enriched in pathways related to response to stimulus, metabolic and development related processes, which is quite within our expectation. So next, we want to further check if this increase of this burn microRNA 6906 can mediate the programming of the growth trajectory and adulthood adiposity induced by paternal PM2.5 exposure. So what we do is we have a normal zygote, which collect the cells, and we inject them with the microRNA, which is the thin-sized one, or a control scramble microRNA, and then we combine them and transfer them to a surrogate mother and produce offspring. We want to check if these phenotypes, the growth trajectory and adulthood adiposity, can be reproduced by this injection of microRNA. So as you can tell from these figures, compared to the control group, the microRNA 6909 treated offsprings have significantly low birth weight, retarded postnatal weight gain, reduced food intake, and increased adulthood adiposity. All of these phenotypes we see or we observe in the offsprings from the paternal PM2.5 exposure, we have repeated. So which means it's very likely that this microRNA is playing a very important role mediating this effect. So finally, we want to check what is happening. So we collected the embryos, the 4-cell embryos of these microRNA treated mice, and we checked. We did RNA sequencing. So what we found is that among the 16 significantly enriched GO terms, we find 4 of them are directly related to the development or the function of neural system. So this is just a indicating us that the increase of the paternal sperm microRNA 6909 level is highly likely program offspring's growth trajectory and adulthood adiposity through targeting the neural system. So this is just a suggestion for us for the further studies. So next step, we might need to further dig into what is happening, how the neural system is playing a role in this microRNA mediated developmental programming caused by the paternal exposure. So just a very brief comparison of what we can see from these 2 studies. From the maternal exposure side, we find the preconceptional exposure, preconceptional period is very sensitive or very important to program the offspring's developmental development and adulthood adiposity. For the maternal exposure, to PM2.5. And we found this programming is maternally transmitted across 3 generations from F0 to F2. And we found the epigenetic modification on leptin expression could be very important in this effect. But for the paternal exposure of the PM2.5, we found that the developmental programming is more like a retardation of the postnatal weight gain, which is quite different or opposite to what we see in the maternal exposure. But similarly, we also found adulthood adiposity in the male offspring. And we also found this effect is multi-transgenerational transmitted. But they are along the paternal line instead of the maternal line, like what we see in the maternal exposure, and they can last it from F0 to F3. And very importantly, we might find a biomarker or very important mediator of this effect, which is microRNA 69095P. So further studies, we're still working on, because there are so many things we need to check. And the real world is usually not about maternal exposure alone or paternal exposure alone. It could be maternal and paternal exposure at the same time. So which ones will stand out, and what is the complex mechanisms when they happened at the same time, or there could be other potential pathways leading to this developmental programming by parental exposure to PM2.5. So we're still working on this, and this is just a brief introduction about what we have done about the cross-generational effect of this PM2.5. So finally, I would like to thank to my colleagues and students in the A group from the Department of Environmental Health in Fudan University, to my collaborator, Professor Yin from University of Maryland. And also thank you for this for all the funding support from these organizations. Thank you. Thank you for your attention.
Well, that is very, very impressive.
Okay, if any of you want to contact me or ask for any information, you can send me email through this email address.
Great. Well, that's a lot of work and very impressive. I'll just start off the questions. I see there's one in the question and answer, but let me start off with a question for you first. And it's a fairly general question, but I presented about ultrafines. You're doing PM2.5. There are a lot of combustion particles, but the one I wondered about as you were presenting is tobacco smoke. And I wondered if any of these same pathways and same findings have been found either in animals with tobacco smoke or in epidemiology like I do with children or second or third generations of smokers, if you know anything about that.
When I check the cross-generational effect, if you're talking about the cross-generational effect, it's not very well studied. As you can tell from the studies, like you have to go through, offsprings, like F1, F4, it takes a lot of time, even though we're using mice models. So it's not a lot of work if you check the literature. But if you're talking about like toxicological mechanisms or pathways of this, the adverse health effects caused by tobacco smoking, you can find a lot.
Yeah, the ambient particulates have a different composition than tobacco combustion particulates, but they're all particulates and they're not good for you. And so it strikes me that maybe that would be a relevant issue. There's a question whether which is similar to mine, but in a different direction. Can PM2.5 cause obesity development in offspring?
Yes, that's what I've is that I did not see the question. Like you're talking about the in offspring, right? So that's what I've mentioned. For maternal exposure, which means if the mother is exposed to PM2.5 during preconceptional or preconceptional gestational periods, the offsprings, which it's very highly likely to be more sensitive to obesity. So that's what we find in mice models. Actually, this is not very new because developmental programming, like what you can tell from epidemiological studies, some of the studies also find that if the mother is exposed to PM2.5, the offsprings might suffer from obesity or other diseases like asthma and so forth. So this is not very new, but the mechanisms or the multi-generation it's transmissible or not, it's more difficult to be done in population, in human. I mean, you need to spend a lot of time. So we use mice models instead here.
More time than I have at my age, I think, to follow people over 3 generations. I do think, though, that, it's interesting. You're doing toxicology lab research. I'm doing mostly epidemiology. We did a couple of we did some metabolome, and we did one study in rats, but mostly it's humans. And we're studying different outcomes, but I do think that there is a very important potential to bring together people like you and people like me to work together, and for somebody to be doing the epidemiology in the human population and someone else to be doing the toxicology in animals, because those two approaches to research have very complementary strengths, almost the opposite strengths and weaknesses. The animals, you can control everything. You can look at all the outcomes. We can't dissect people's livers out, right? So you can do that in a mouse, but it's a mouse. It's not a human being, and there'll always be an asterisk next to it. Does this happen in humans? With humans, they're humans, but so many things are going on. You saw what we had to do just to assign exposure to people because they're moving. We don't have any control over them. They're doing all kinds of things. There's all kinds of factors interfering, and I think there's potential for epidemiologists and toxicologists like you and me to work together iteratively to strengthen our confidence that we know what's going on in human populations. So I just say that I've thought that for a long time, but it comes back to me listening to your talk and thinking about what I presented, and also imagining people listening might think, well, what's the connection between these two? But I think there is an incredible importance to have both sides of this work going on.
Yes, I definitely agree with you. I always think if my so on one side is like what you find in the population or in epidemiological studies. We want to know if we could get rid of the other factors, like for example, like what you said, other pollution from other pollutions, effect from other pollutions, or if there is any effect of like diet or other things, and we can well control our exposure and also well control the diet and other factors you can think about in mice models. And personally, yes, I always think if my work we can collaborate with epidemiological researchers, and we try to understand very interesting topics. And personally, I'm not just working on, as I have mentioned, I'm not just working on the cross-generational effects. I also work on a lot of the action modes of the particulate matters, how they translocate in vivo, of course, using animal models. And of course, as you said, like the size effect of these particulate matters, the chemical effect or the shape morphological effect, that's what I'm interested. And for my lab, we're not just having this PM2.5 exposure, whole body exposure system. We also have the ozone exposure system for animals. I mean, because I'm working on animals, and we are also working on exposure system for ultrafine particles. And we have a lot of in vivo tracking systems to track what is happening inside the mice. This could be done in mice models or animal models, but not in humans. So there's a lot of things. Of course, I think there's a lot of things can collaborate, combining these two sides and work on maybe a little more deep to understand what is happening about the effect of these particulate matters in the body. So yeah, that's what we have been done in our lab.
That's great. Are there any other questions from the people who are still listening? If there are, there's a little bit of time. I actually have to drop off in about 5 or 6 minutes because I got scheduled back to back all day today. So I have a 9:30 my time here on the East Coast of the United States. But we maybe have time for 1 or 2 more questions if there are any. If not, I don't know, Cedric, do you want to say something as a closing to the session?
Well, if you don't have any closing words.
Well, I would just say, Dr. Zhu, it's a pleasure meeting you. I really enjoyed doing this with you. I couldn't tell who all the people listening in were, but they seemed like they were from all over the world, which was really pretty cool. And I like that. Let's keep in touch. Maybe there's something we can collaborate on in the future. And then I would just remind listeners that we have this special issue of the journal Toxics. Please consider making a submission to it. It's about particulate matter and biomarkers. Could be epidemiology, could be toxicology, could be either. So far, I think most of the papers that are coming in are epidemiology. But thank you. So I know it's evening there, right, Dr. Zhu? I don't know what time.
Yeah. Yeah.
But I appreciate you staying up and giving part of your evening over to this. That was nice of you. And Cedric, you're the one who has the best time of day of all of us, right? You're in what, Switzerland?
Yes, exactly. It's 3:20.
How come you get the good time? But anyway.
I don't know. Yeah, well, maybe just a few more words from my side. I would like to thank our speakers for their time and interesting contributions to this webinar. Also, thank you to our attendees. I hope you found this webinar interesting. As a reminder, the recording of the webinar will be made available as soon as possible on the webinar's webpage. And to access your certificates of attendance, please log into your SCI forum profile or create an account with the same email address you used to register and attend for the webinar. The certificates are available in around 30 minutes. And yeah, now I wish everyone a nice day, morning, evening, wherever you are in the world. Take good care. Bye-bye.
Okay, great. Thank you, everyone. Have a good, again, you said it well, morning, afternoon, evening.
Yeah.
Bye-bye.
Have a nice day.
Take care. Bye-bye.